A very big hello to all my wonderful PU2 champions. I hope my dear friends are doing absolutely good. I'm Jasmine and I want to tell you one very important thing before I begin up, and that is, if you are someone who do not have much gap for biology, that is, you might have mathematics in between or Hindi in between and you're worried about that, please do not be worried. Worrying would not help. It will worsen up things. It will not help your preparation, right? Yes. So what we have to do? We just have to work. So worry less and work more, and I am here with you throughout. I'll tell you what's important. I'll tell you the shortcuts also. Well, there is no big shortcut for biology, but there is a way where you can complete everything that is important. Okay? And these are the most important 5-marker questions that I have sorted out for you. I went through all the MQPs out there. I went through question bank. I have curated questions. I have put it over here, such that there is no way that you can lose marks in the 5-marker session. And if you get all the 5-marker questions correct, how many marks do you get? You get 25 marks, because you can attempt 5 of the 5-marker questions. So 5 into 5, 25 marks. And that itself is a very big deal, right? Yes. So make sure you give me a promise right away that you will solve each and every question. Not think it's too big and things like that. No, it's just some parts that you're going to see over here. It is not going to be big. I'm telling you, trust me on this. Okay. So promise me one very important thing, that you will be solving each and every question. And if at all you promise me, put it down in the comment section. No, the spelling is somewhere wrong. Promising you for 70 on 70. Our target is going to be this only, and you have to promise me that in the comment section. Okay? And yes, we'll go ahead, we'll solve some questions, we'll see some questions, and I'll tell you where you can find the solutions for it as well. Okay? So these 5 chapters does not give any 5-marker questions, that is reproductive health, evolution, microbes and human welfare, ecosystem, biodiversity and conservation. They only have 2 and 3-marker questions, which I have already discussed. If you have not watched this video, you can watch that particular video as well. Now, there is a way where I can give you the most repeated fixed questions. That is this way, where I just give you topics. It looks very small, very easy to cover and things like that. But do you think we are here for something that will make our work super easy and not useful? No. So basically, I have put the same questions in this format. What do you mean by that? This is in question format. I have given you a question on double fertilization. Now, they have put the term double fertilization, but here you see an actual question in a different way. And this gives you an idea about how a concept can be asked in a different way and how can I get my whole marks. So as I am telling you, I do not want any comments in the comment section saying that, oh my God, there are so many questions. No, there are not so many questions are there. There are limited questions, and these questions are definitely going to help you out as well. So according to your MQP, when I studied, I get 3 topics from where you have more chances for the question to come. One is double fertilization. One is the gametophyte, as in the female and the male gametophyte, which is pollen grain, as well as your embryo sac. And then you have outbreeding devices. So these are the things that can have more chances. There's a chance factor over here. No one can tell you what is exactly fixed, right? But again, I have made a list of the questions in a format that you can practice and try it out and see, are you able to answer these questions or not, because as I said, there are chances for that to come. This is from a question bank. I've not taken from anywhere else. It's from your question bank that is the government had given. So make sure you follow it. Yes. Now the next thing is human reproduction. Again, these are the questions which are there, and now I want to tell you one very important thing. In 5-marker, any of this diagram have a short chance to come. First thing, this is their first priority, and then comes all these questions out there. Okay, so this is the first priority, that is human reproduction system, male reproductive system, mammary gland, schematic representation of spermatogenesis, or basically it can be oogenesis also. Any of this have more chances to come. A diagram-based question has more chances to come in human reproduction. And over here, these are the other type of questions which are there. These are from the question bank. These are hot question which I want you guys to solve. I have solved it. I will be showing you the video also where you can find the solution for it. This is a question that is new. Okay, I have not solved it anywhere, but you can try it for yourself, and I'll be coming live also for helping with all this. So do not be worried about anything. Any question that you're not able to solve, ask me in the live. I will be there to help you with that. And by the way, if at all you are someone who have not subscribed to the channel, you're missing out on all the live updates, all the very important information, make sure you click on the subscribe button right now. Yes? Okay. So I hope this is clear. This is short question. I have done a separate video on most important diagrams. Watch it. If you're if you're struggling with the diagram making, if you're okay with diagrams, you do not need to watch it. Do not waste your time. Okay. So I do not want you guys to waste your time. It's not for views. I am here as a teacher, as just a teacher. Okay. Principles of inheritance and variation. You have these many 5-marker questions which are there. So make sure you try it up, and there is high chance for one question. So in principles of inheritance and variation, you are going to get 2 5-marker questions. You're going to have an option of having 2 5-marker questions. So one chance is there for the schematic representation related questions to come, as in basically monohybrid cross, that is known as 1 gene inheritance or 2 gene inheritance, or basically incomplete dominance. These are things that there are high chances for that to come, or basically you can get questions related to the dihybrid cross, monohybrid cross, and things like that. So I've given you this question as well, okay, for you to practice. So either this way or in this way, you can get a question on this. And apart from that, genetic disorder related questions, such as you might be given some symptoms, you have to find out which is that particular disease, or you can get questions based on this where you have to make a pedigree chart and things like that. So anything related to genetic disorder can also have high chance to come. The unique questions, I have put it over here such that you can also have a go through of things. Okay, now this question I have not solved before. If you find difficulty, I will be there in the live solving these questions. Okay, do not worry about anything. So yes, most of the questions, there are questions I have taken from MQP also, which gives you an idea. Apart from that, yes, these are the new questions which are there. Cool, let's go ahead. Now, we have molecular basis of inheritance. These are the questions which are there, my dear children. Now, lac operon is something where you can get question from transcription and RNA polymerases. Different type of transcription. What is the difference between prokaryotic and eukaryotic transcription? What are the different form of RNA polymerases enzymes in eukaryotes? What do they transcribe? Each of them, there are 3 RNA polymerases, so you please know about that. Now the search for DNA, the experiments, that is Griffith experiment, Hershey and Chase. So Griffith is given in your MQP, probably, which makes an understanding that if at all you're pro in that, please make sure you go ahead with Hershey and Chase also. There are high chances for that to come. This question can be asked in a different format. Which experiment gave the final proof for DNA as a genetic material? It was Hershey and Chase. So please know questions can be given in different format. Different types of thing can be asked to you as well. Radioactive sulfur and radioactive phosphorus get incorporated into which parts of the viral particles. So please know about the basics. Yeah, and then genetic code related questions can also come. I have put all the questions over here in a format that you can help with, and some extra questions also, just for your safety. So yes, now name the process involved in the following. Again, I have given you the answers over here. These are the questions which was asked. I've given you the answer. This looks very big, no? But the answer is two things only. So that's what I am trying to tell you. Don't be scared of the look of the question. Now we have human health and diseases. These are the questions which are there, my dear children. The life cycle of the missing data of either of them, either HIV virus life cycle, they can give you missing data also in between, or probably the plasmodium's one. This is a very important thing. Make sure you go through it. And apart from it, cancer and immunity related questions have more chances to come. Immunity related as in acquired and humoral immunity, acquired and innate immunity and things around that. Okay, there are more chances for that to come. Now name the group of genes that have been identified in normal cells that could lead to cancer. All these questions have been put down. This is a new question that's there. The picture for that is, just a second. The picture for that is rightly put over here. Okay, so name of the disease, causative organism, and symptoms are given over here. So questions based like this can also be coming in. Yes. So, but these have more chances. So first priority them, then anything else. Okay, biotechnology principle and processes, again PBR 322, you know it's important. Anything related to that, the working of PBR 322 can be coming in. So yes, the transformants, recombinants, these terms are super important. Know the difference about it. If not, you can watch my video. I have explained in detail. Now the tools and the process involved has more chances to come apart from PBR 322. Tools as in basically what all do you need, restriction endonucleases, this and that, DNA ligase and vectors. Okay, so all the different type of vectors are there, and the processes which are the gel electrophoresis, what the stain is the one that you use and things like that. So these are things that can be coming in. Make sure you go through it. Apart from that, there are new questions that I have put up. Yes, try to solve it from your end. Okay? Yes. Now, biotechnology and its application, these are the questions which are there, my dear children. Make sure you go through each of them. Over here, I was not able to deduce what's more important because in 3 MQPs, 3 different things were being asked. In one, transgenic animals, in one, genetically modified plants, and in one, it was asked about the biotechnology in the field of medicine. So Yes, so anything related to this or apart from this, for example, RNA interference can be asked, Bt cotton working can be asked, which is why I didn't put anything over here as a topic that you have to go first. Make sure you go through the entire thing, because these chapters, biotechnology and its application and biotechnology principle and process, gives only one 5-marker question, and then only the MCQs are there. So you have to grab those 5 marks. If you're studying those chapters, you have to grab those 5 marks. Yes, yes. So yeah. And organism and population, this would be the last chapter that we are going to discuss, because that is the only one that gives us 5 marker now. Now over here also it's very difficult to understand what is more important. So these type of questions have high chances to come. What is mutualism? All the interaction based questions, or basically some question like this where you have to fill in the blanks. So they have given you a question on biological interaction in the MQP. Similar type of question can be framed like this. So you should be knowing all the blanks are there. Apart from it, this curve related question, the growth curve related question can also be coming in. I have discussed all of it already. In your MQP, you have population density based question also. So please go through the advanced questions that I have mentioned over here. It will be really, really good for your practice. Okay? Yes. And the answer for all of them, most of them, is basically in the 5-marker question video. Again, I'll put the playlist in the comment section or things like that, where you can find the 2-marker, 3-marker, 5-marker also. Apart from that, I'll be coming live, I'll be solving it. But if you're someone who feels that I do not study well in live, I want to have a recorded session, I want to utilize maximum amount of your time, I do not mind. Okay, I do not mind. You can go ahead and watch this, but make sure you practice the extra questions also, which I have marked over here. I marked it as extra. Why? Because I want you guys to practice it, right? Yes. So make sure you do that, and I hope this video had helped you. If it did, definitely let me know in the comment section. And my dear children, I am here for you guys anytime. Okay, remember that. And do not be worried, we'll do it. Okay, we both will do good. And if at all you are someone who is interested in KCET, we have launched KCET crash course. It's online crash course which is available for rupees 2499. It's known as KC30. You can go enroll into it because there's an offer going on. The price is reduced. Similarly, if you're someone who want to have an offline version of it, you have Lakshya KCET crash course. 3,000 off is ongoing. You can check it out. And if you're just worried about having a good test series and that's all, you can avail it for rupees 999. That is ABHYAS KCET test series. It's for those who are going ahead with KCET. Okay, if you're not going ahead, then chill pill. Now, wishing you all the best. Please take care of yourself. Bye-bye.